Chloroquine, a name synonymous with malaria treatment, has been a global health champion for decades. This inexpensive and widely available drug has been effectively fighting off the parasitic disease, offering relief to millions worldwide. But the story of chloroquine doesn't end there. Scientists and clinicians, in their constant pursuit of innovative cancer therapies, have turned their attention to this old drug, uncovering its potential in a new battleground, the fight against cancer. This repurposing of existing drugs is not a new concept in medicine. It presents a faster and often more cost-effective way to develop new treatments. The familiarity with chloroquine's safety profile and its established production processes makes it an even more attractive candidate for repurposing. Its potential to target cancer stem cells, a particularly aggressive and treatment-resistant population within tumors, has ignited excitement in the oncology community. While traditional cancer therapies effectively kill the bulk of tumor cells, they often struggle to eliminate these elusive cancer stem cells. These stem cells, with their ability to self-renew and differentiate, are thought to be responsible for tumor recurrence and metastasis, making their eradication crucial for long-term remission. This is where chloroquine steps in, offering a potential solution to this persistent challenge in cancer treatment. Cancer cells, particularly cancer stem cells, are characterized by their high metabolic demands and reliance on a process called autophagy for survival. Autophagy, meaning self-eating in Greek, is a natural cellular recycling process where cells break down and reuse their own components to maintain energy balance and manage cellular waste. In cancer cells, this process is often hijacked, becoming a survival mechanism that allows them to withstand the harsh conditions within the tumor microenvironment and resist the effects of chemotherapy and radiation. Chloroquine disrupts this critical survival pathway by inhibiting autophagy. It achieves this by preventing the fusion of lysosomes, cellular organelles responsible for degradation, with autophagosomes, the structures that engulf cellular components destined for recycling. This blockage leads to an accumulation of waste products and damaged organelles within the cancer cell, eventually leading to its demise. Furthermore, recent studies have shown that chloroquine's impact extends beyond just inhibiting autophagy. It appears to modulate various cellular signaling pathways involved in cancer cell growth, proliferation, and resistance to treatment. This multifaceted approach makes it a promising candidate for combination therapy, where it can be used alongside existing treatments to enhance their effectiveness and potentially overcome drug resistance. The potential of chloroquine as an anti-cancer agent was first recognized in the early 2000s, sparking a wave of research to explore its efficacy and understand its mechanisms of action. One of the pioneers in this field, Dr. Dong-Soon Choi at Seoul National University, demonstrated the drug's remarkable ability to target and eliminate breast cancer stem cells. His groundbreaking research, published in the Journal of Clinical Investigation in 2008, revealed that chloroquine, when combined with conventional chemotherapy, significantly reduced tumor growth and prevented metastasis in mice models of breast cancer. Another significant contribution came from Dr. Steve Pascolo at the University Hospital of Zurich, who investigated chloroquine's impact on glioblastoma, a highly aggressive brain cancer. His team's clinical trial, published in Autophagy in 2010, showed that adding chloroquine to the standard treatment regimen for newly diagnosed glioblastoma patients resulted in improved progression-free survival. These promising results, though from a small trial, provided the much-needed impetus for further clinical investigation of chloroquine in various cancer settings. These early studies, along with numerous others, have solidified chloroquine's position as a potential game-changer in cancer treatment. They have not only provided evidence of its anti-cancer activity, but also shed light on its mechanisms of action, paving the way for more targeted and effective therapeutic strategies. Section 4. A wider net chloroquine's reach across cancer types. The excitement surrounding chloroquine's potential is not limited to breast cancer or glioblastoma. Preclinical studies and clinical trials are exploring its efficacy in a wide range of cancers, including but not limited to triple-negative breast cancer, pancreatic cancer, leukemia, and melanoma. These cancer types, known for their aggressive nature and limited treatment options, make promising candidates for chloroquine-based therapies. Triple-negative breast cancer, for instance, lacks the expression of 3 common receptors that are targeted by hormonal therapies, making it particularly difficult to treat. Early studies suggest that chloroquine, by targeting cancer stem cells and enhancing the effects of chemotherapy, could offer a much-needed therapeutic avenue for this aggressive subtype. Similarly, pancreatic cancer, notorious for its resistance to conventional treatment, has also shown vulnerability to chloroquine in preclinical models. The potential of chloroquine, however, extends beyond directly killing cancer cells. Researchers are investigating its ability to modulate the tumor microenvironment, the complex network of cells and molecules surrounding the tumor, which plays a crucial role in tumor growth and metastasis. By influencing the immune response and reducing inflammation, chloroquine could further hinder tumor progression and improve treatment outcomes. Section 5. Striking a balance. HCQ, a safer alternative. While chloroquine holds immense promise in cancer treatment, its long-term use, especially at higher doses, has been associated with potential side effects, particularly retinal toxicity. This risk has prompted researchers to explore safer alternatives, with hydroxychloroquine, or HCQ, a derivative of chloroquine, emerging as a frontrunner. HCQ, commonly used to treat autoimmune diseases like rheumatoid arthritis and lupus, shares a similar mechanism of action with chloroquine, but is considered to have a better safety profile, particularly with regard to retinal toxicity. Numerous clinical trials are currently underway, evaluating the safety and efficacy of HCQ, both as a single agent and in combination with other therapies in various cancer types. Early results from these trials have been encouraging, demonstrating HCQ's ability to inhibit tumor growth and improve patient survival with a manageable side effect profile. Its established safety record and widespread availability make it an attractive option for further clinical development as an anti-cancer agent. However, it is crucial to remember that even though HCQ is considered safer than chloroquine, it is not without risks. Careful patient selection, dose optimization, and regular monitoring for potential side effects remain paramount to ensure patient safety throughout treatment. Section 6. Vigilance is key monitoring for potential side effects. As with any medication, chloroquine and HCQ come with potential side effects, and it's crucial to be aware of these to ensure patient safety. While generally well tolerated, especially at lower doses, chloroquine can cause gastrointestinal issues like nausea, vomiting, and diarrhea. These side effects are usually mild and manageable with supportive care. The most significant concern with long-term chloroquine use is retinal toxicity. This risk is dose-dependent and can lead to irreversible vision loss if left unmanaged. Regular ophthalmological examinations are therefore crucial for patients receiving chloroquine therapy, especially at higher doses or for extended periods. Early detection of any retinal changes allows for prompt dose adjustments or treatment discontinuation, minimizing the risk of permanent vision impairment. HCQ, while considered safer than chloroquine, can also cause retinal toxicity, albeit at a much lower rate. Regular eye exams are still recommended for patients on HCQ therapy, especially those with pre-existing eye conditions or other risk factors. By closely monitoring patients and implementing appropriate preventative measures, healthcare providers can mitigate the risks associated with these drugs and ensure their safe and effective use in cancer treatment. Section 7. The future of cancer treatment, a hopeful horizon. The emergence of chloroquine and HCQ as potential cancer therapies, particularly their ability to target the elusive cancer stem cells, marks a significant step forward in our fight against this complex disease. While challenges remain in terms of optimizing treatment regimens and managing potential side effects, the future of chloroquine in cancer treatment appears promising. Ongoing research is exploring its potential in a wider range of cancer types and investigating its synergy with other therapies, paving the way for more effective and personalized treatment strategies. Furthermore, the development of novel drug delivery systems and the identification of biomarkers to predict treatment response hold the potential to enhance its efficacy and minimize toxicity. The journey from an anti-malarial drug to a potential cancer therapy underscores the power of scientific curiosity and the importance of exploring existing resources for new applications. The story of chloroquine in cancer treatment is still being written, and with continued research and a focus on patient safety, it holds immense promise in improving cancer treatment outcomes and offering hope to millions battling this devastating disease.